So to introduce myself, for those of you that don't know me, my name is Thor Rollins. I work at Nelson Laboratories. Nelson Laboratories is a CRO that does preclinical testing for medical devices. Part of that medical device testing is biocompatibility, which sometimes is like a four-letter word in some people's mouths. But that's kind of what we're here today, is to kind of talk about biocompatibility. And it's a very hot topic today, or right now in the industries, because there's a lot of changes happening regarding biocompatibility. Not so much the testing or the endpoints, things like that, but how they are applied in the regulatory agencies, and kind of the approach that we need to take for biocompatibility. So along those lines, just recently, in fact, in June of this year, the FDA released their new guidance document on biocompatibility, and that's what we're going to kind of talk about today, is what's in that guidance document, what changes are going to impact the medical device arena with that guidance document, and what can we expect going forward. How's it going to be implemented, what we've already seen implemented, things like that. Oops. So first off, to kind of let you know, we're going to go through a very quick background with FDA guidance documents on biocompatibility. We're going to talk about that draft that surprised everybody in 2013, and then how that draft became finalized just earlier this year. So that's kind of the approach we're going to be taking today. So what kind of started in biocompatibility was in 1995. The FDA released a document called the Blue Book Memorandum, or the Bible, so to speak, of what biocompatibility was. And it was a really good document. It went through what risks were associated. It mirrored ISO 10993 at the time. And so it was a good guidance document. Unfortunately, 1995 was a pretty long time ago. And so the FDA slowly started to change how it approached biocompatibility, and there wasn't any kind of written guidance on what that meant. And so people would submit to the FDA, they would get feedback, and they would have to change how they would do biocompatibility. And so the next time they submitted, they would do what they was told last time, and lo and behold, something had changed again. And so it was kind of this moving target that we were constantly trying to hit. And at Nelson Labs, I mean, we interact with the FDA on a daily basis, and it was still difficult sometimes for us to hit that moving target. So we saw some signs of things changing. FDA is very active in the TC194, which is the working group that writes the ISO 10993 standards. And so, and when we were on that standard committee, we saw the FDA's input, and we kind of got a good idea. Things were starting to get more defined, and the programs are starting to become better situated. With that, okay, so this was this is out of the ISO 10993, but more specifically was out of the G95. And for those of you that grab these on the back, this is just a slide rule of this chart, because we found that engineers like to slide things. So we have made this chart into a slide rule. But this was what really was the risk evaluation for medical devices in the old ISO 10993 and the G95. Basically, what you did is you evaluated the contact type of your device, how long it was in contact with the body, and that told you what your risk was. And by risk, I mean it told you what testing to do, right? So it was a checkbox mentality. You just went through whatever testing it told you to do here, you did that testing, you submitted to the FDA, and that was your biocompatibility. So the guidance in the standard told you what the risk of your device was. And that might have worked better in 1995, but really devices have become much more complex, have become much more hard to assess the risk just by putting it into some simple categories. So it kind of called for something to change as far as how we evaluated biocompatibility. For those of you that didn't get one of these, there might be some more back there, I don't know. But if not, come by our booth. We have more of them, and so we can hand them out then if you did not get one. So with that, with the changes of the medical devices, in April of 2013, the FDA released a surprise draft. And what I mean by surprised is we really know it was going to happen. In fact, we were meeting in Italy as a 10993 working group, and it was just happened to be when the FDA was sequestered. So the FDA couldn't send their representatives to this meeting, and so we thought to get us back for us being in Italy, they submitted this draft, surprise draft. So it kind of hijacked the meeting for a day where everybody was reading this draft document, trying to understand what was different. But it was great. It was a very good draft of what the current thoughts of the FDA were at that time. And then the FDA allowed 6 months for comments. And they got back hundreds and hundreds of comments from this draft. So much so that Jennifer Good, who kind of oversaw this process, she showed me the red line version of the draft, and it was completely red almost, because how much changes they had to put into it. Because of that, it took quite a long time for them to go from the draft phase to a final document. So with that, the final document was released in June of this year, and then became really effective in September of this year. This draft actually is a very good product. I think it's a very useful for medical device manufacturers, and it's longer than the other drafts, so it could take a little bit more time and coffee to get through. But I would recommend getting through this draft because it really does give you insights into what the FDA is thinking as far as biocompatibility. We are going to hit the high topics today, so but I would recommend still reading the draft because it does flow really well and give you that kind of insight. But it was very different from the draft document, this new guidance document. It had changed in some aspects where we had comments. It was more detailed. There's a lot more invested into this document than the draft document. So this is the draft document, some of the aspects that we like to talk out. But the main thing I want to talk about now, the big change that we saw from the FDA is all about risk. No longer is this checkbox the only thing that you do to evaluate risk for your device. You have to understand your device now, which is kind of a harder thing to do. But you have to evaluate your own risk to the FDA. These endpoints that this chart gives you are still endpoints you need to assess. So it still gives you a good guidance on what needs to be assessed for your device. But it's no longer a checkbox, black box kind of mentality where you just check, 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 check. You have to prove to the FDA that these endpoints are appropriate for your device and why you think some of them may not be. So it's a unique classification per device. So risk-based approach is what we're going to hear. In case, and if you guys want a copy of this presentation, just leave me your business card afterwards. I'll get you a copy. It doesn't matter. But this is the link for the draft document, or you can just put in Google FDA guidance document biocompatibility, and it'll be the first thing that pulls up. So what we've done today is we've broken down this new guidance document into 3 categories. The first category of summary of ideas. These are ideas that we've kind of seen implemented throughout the process at the FDA, but now we have them implanted into a document. We have justification ideas. So FDA gives us thoughts on how to justify out of testing. They no longer want you just to checkbox, right? So they give you thought process on how to evaluate the risk and justify why some testing may not be necessary. And then we've seen some things that have maybe big impact in the medical device arena and have already had big impact in the medical device arena. So these are just some high-level things that we're going to go through really quick. I've dug out some more impactful things later on that we're going to spend some more time on. But there's some things in there about fluid or gas path devices. Let you guys know if you have a fluid or gas path device, so especially gas path, this is something that's is implemented in the last couple of years in the industry. That gas is considered a liquid extract that can pull chemicals and particulates from the material and deliver it, especially if it's inhaled, into the lungs. Okay, so inhaling particulates really is not a great thing from a toxicological standpoint. Plus, a lot of inhalation toxicity has a very sensitive endpoint depending on the compounds. So any kind of liquid or gas path devices the FDA has particular concerns about and may have to appropriate chemistry test to evaluate that impact to the user. They also have incorporated different standards into this. They've pulled out ASTM standards. They pulled out the different test-specific standards for references. And they hit home again that the FDA does not approve individual materials. I can't tell you how many times I hear this when I talk to clients. My device is made out of this polypropylene that's medical grade. Why do I have to do biocompatibility? Or we've used this for years. Why do we have to do biocompatibility? These are great questions to evaluate risk, but you can't just evaluate a material by itself because that's only half of the device. The other half is the processing that you do to it. The residuals from cleaning, the residuals from the manufacturing line, the sterilization impact on those materials. So there are a lot of things that go into your risk assessment besides the materials that you have to evaluate. Material is a great point to look at, but it's not the only stop point. You have to go into more to evaluate the risk of the device. That goes along to master files too. If you have a silicone that has a master file with the FDA and you think, I'm done, no biocompatibility for me, that's not the case, okay? First off, not all master files are made the same. So the information that's provided to the FDA in the master file may not be sufficient. Secondly, once again, you're forgetting about the processing of that material. So you have to evaluate that endpoint too. In fact, in the new guidance document, the FDA gives specific instructions on master files, what should be included when the people are submitting master files for materials. Permanent contacting devices. We're going to talk about this later on, but just remember that if you have a permanent contacting devices, that chronic toxicity and carcinogenicity endpoints that we've always forgot about because they're uncomfortable when you look at a price tag or turnaround time are no longer something we can just forget about. And we'll explain that in a little bit. And then device failures. If your device is going to possibly fail during use and expose part of the body that was not intended, we have to address that failure now too, which was always kind of the case, but the FDA really specifically spells it out. One is something that we're still trying to talk with the FDA on as far as what the impact is, but it revolves around the test of cytotoxicity. And not to get into too much detail, but I could because I love this test a lot, but you guys probably don't, so I'm not going to get into too much detail. But this test, we extract usually in history, and if you look at the ISO standard, it says to extract the samples at 37 degrees for 24 hours. And it has to do with an L-glutamine portion in the media where it's not really heat stable. So the longer you extract it at a temperature, the more it degrades. So it allows you to only extract for 24 hours. The FDA has come out in this guidance document and says if your device has longer than 72-hour contact with the body, it wants to see a 72-hour, 37-degree extract in the cytotoxicity test. This test, so we still pass this endpoint at 37 degrees for 72 hours, but it may make the test more sensitive. We're still trying to determine if you get a 1 or 2 by using a 24-hour extract and now you're extracting for two more days, how does that affect the sensitivity of the test? I think it has a pretty big impact, actually, but we're doing some testing now internally to try to determine if we can really have an impact of this longer extraction period. If you have any type of circulating blood contact, you have to do the total hemocompatibility tests. We once again have to evaluate risk, but it is something that we have to look at. And then they talk about implantation being a muscular preferred over subcutaneous. And if you think about how we usually do an implant test, it's in a rabbit. The rabbit's subcutaneous tissue is a little loose, right? And so intermuscular helps control the device and also have a more sensitivity because of the interaction with the muscle tissue in the surface of the device. Now some of the justification ideas that we can use. The first thing that you want to look at is the risk of the device versus the benefit of the device. And this has been something that the pharmaceutical industry has used for a long time, but it's something that we can start really evaluating of medical devices. For example, if I have a device that's going to prolong someone's life 20 years and it causes a rash, well, maybe we should still have that device on the market and just deal with the rash and not have that person's lifespan be shortened, right? So we can look at the benefit of that device versus the risk that we see with the biocompatibility. It allows us to maybe be a little bit more creative with some of the failures we may see from biocompatibility testing. We can also look at literature endpoints, specifically when we start looking at chemistry. And we'll talk a little bit more of this in a minute when we start talking about chemistry. But literature review of endpoints of compounds based on how the device is used may help us justify out of some of this testing. And this goes to when you look at toxicity endpoints, carcinogenicity endpoints, genotoxicity endpoints. We can find literature studies to help us justify that those tests do not need to be performed. And in fact, we've been very successful in the last 6 months doing that, justifying out of those endpoints. And in fact, those are the endpoints I have listed down there that help us with that. I do want to put the idea into your minds, start thinking about chemistry as a synonym to biocompatibility. Okay? Extractable leachables can help evaluate risks of a device and help eliminate certain endpoints listed down below, where we can then evaluate those endpoints using chemistry and not have to do the tests. That is something the FDA wants us to look at, something they put in their guidance document, something they've stressed to us on the phone, something that's been very successful. And, by the way, it happens to be cheaper and quicker than some of these animal tests, and to get better information. Okay, so functionality tests. How many of us use certain type of functionality tests at the beginning of the design process to evaluate how the device performs? If we do that in an animal, talk to us first, because I'd be more than happy to give you endpoints that we can put into those tests to evaluate biocompatibility. A lot of times, biocompatibility is not evaluated into the functionality endpoints, and we can help with that. If you just contact us beforehand, even if we're not doing the testing, I'd be happy to work through some endpoints that we can put into those protocols to help you evaluate biocompatibility. It just helps save animals in time and money. And predicate devices can help limit testing if they're appropriate predicates. I do have to be aware here that a predicate device is just not materials, right? We talked about processing too. That's sometimes harder to do unless you guys manufacture that predicate. But if we can associate predicate device as far as materials and processing, this is great for a change of a device. If you're changing a device, let's look at the old device as a predicate device and see what kind of impact it may have. We can reduce the amount of biocompatibility testing that needs to be performed. So when we start looking at some possible impacts, and like I said, we'll get into a couple of these in more detail in a minute, but the first thing is practitioner impact. Okay? This was the first thing that if you read the document, it kind of talks about, which is practitioner contact. Practitioner contact is really, and we'll talk about it in a minute, but it's really about those users of the device. It's no longer the risk of just the patient, but also about the people using the device and how that may impact the biocompatibility of that user. Risk-based decisions. It's all through the document. You have to evaluate your risk to the patient before you can do the testing. The testing is designed to mitigate the risk of the initial thing, the initial device. So come up with a plan. Evaluate your risk in a plan, a written document that you can even give to the FDA to help evaluate that risk. Once you have the risk evaluated, do the testing, and then do a BER, a biological evaluation report, and that's in the standard or in the document, that helps sum up all the risk that was evaluated and how the results of the tests sum up that, mitigate that risk. This is what the FDA would like to see. Okay, so once again, recommend submitting a testing plan to CDRH before testing. When I talk to my FDA friends, this is what they tell me to preach to my people that I talk to. They love to be upfront in the process. And sometimes these FDA reviewers in the past have been known as these like bad wolves, right? We don't want to talk to them. We want to have as limited contact as possible. They're great people, and they want to help you get this device on the market. So come up with this plan. We call them BEPs, Biological Evaluation Plans. And it spells out what the risk of the device is, what the materials and processes are, what endpoints you need to assess, how you're going to assess those endpoints, come up with a conclusion. You can submit that in a Q-sub or a pre-sub with the FDA and get really good, helpful feedback. Then you don't have to do unnecessary testing. We can be very selective on how we approach those endpoints in that pre-sub and get real good feedback. And then you know you're not over-testing, or you may not be under-testing. You know exactly where you're at. And this has been a very successful process for us when we've been able to do it. So there's the biological evaluation report. The FDA wants to have that report submitted. No longer can you submit just a package of reports, of biocomp reports from a lab, and say, this is my biocompatibility. The reviewers don't want to have to go through all those pages and pages, and I make those reports, and I don't want to go through those pages by pages, right? So give them a biological evaluation report where you sum up the risk, sum up the results, and get a conclusion, and then even can reference where the reports are. It helps speed up the process, and a happy reviewer, kind of like a happy wife, happy life, a happy reviewer, happy submission, okay? If you don't make them painful, they'll like you, and it'll be easier to get through it. Okay, we talked about cytotoxicity already. The other thing that's changed, if you have blood contact with, your device has blood contact, it used to be that you had to look at two proteins for complement, a C3A and a SC5 beta 9. That has changed. Now you just have to look at C3A protein. The SC5 beta 9 is no longer required, which is good. It means cheaper testing. So everyone loves that, right? Then we have pyrogenicity. This endpoint has been a little bit of an issue going forward for the FDA. So for those of you that don't know, a pyrogen is something that causes a fever, okay? So there are two types of pyrogen tests we run. We run something called a bacterial endotoxin test, or BET, sometimes called an LAL test. That looks for something called endotoxin, which is in gram-negative bacteria. It's only in gram-negative bacteria, and so we have to screen for that type of endotoxin. And we do that by using horseshoe crab serum, which is a kind of a cool test. If you want to come to Nelson Labs and see us run it, we don't actually have horseshoe crabs running around, but we have their serum, which is kind of cool. So you can come and check it out. But we have to run this test looking for that specific bacterial endotoxin. Then we have a test called material-mediated pyrogen, which looks for basically everything else that could cause a pyrogen. It's kind of vague. It's an umbrella test, but it's not specific enough to look for those pyrogens. The pyrogen has to be looked at using those horseshoe crab tests, where the rabbit test looks for just a bucket of everything else that could cause a pyrogen. In the past, the BET test was only ran for devices that had certain contact, like cerebral fluid, or vascular, or ocular contact. But now, according to this document and another guidance document the FDA released, if your device is considered a permanent implant device, they are requiring a BET test. Okay? Now, the BET test is not very expensive. It's cheap and quick. At Nelson Labs, it's like $120, and we can do it in 24 hours. But it's the samples that can cost a lot of money. If you're an orthopedic device and you make an implant like a knee, the test isn't expensive, but submitting knees into a test lab for a lot release test can be quite expensive. So it's one of those impactful tests that we're still looking at how the FDA is going to implement that going forward. Okay, so genotoxicity, going back. In the past, with carcinogenicity, and we'll talk about this also in a little bit more detail in a minute, has been addressed more by just forgetting about it. Okay, so if you had a permanent implant, and in this slide rule, it tells you to do carcinogenicity, you may put a blurb in there that we did our genotox and it passed, so we don't have to worry about carcinogenicity. Most people just didn't do the test. Just so you guys know, the carcinogenicity test is like, $1.1 to $2 million. It can take up to a year or a year and a half to do. Right? Is it higher than $2 million? Thanks, Larry. 2 years. See, Larry knows he does more of those carcinogenicity evaluations. So 2 years. So it's an expensive, long test. No one does that for most medical devices, right? But now the FDA is requiring you to evaluate that endpoint. Does that mean you have to go out and do the animal tests? Oh, I hope not. Well, I'm a lab, so maybe I hope. No, I don't want to do that test either. So what the FDA allows you to do now is evaluate that using chemistry instead of the genotoxicity, where genotox does not really endpoint carcinogenicity. It has a portion of it, but it doesn't tell you the full tell. So the FDA, if you have carcinogenicity marked on this chart, be prepared to do some kind of carcinogenicity evaluation. That may be a material assessment if your device is simple enough and you can make that argument, or you may need to do chemistry to evaluate carcinogenicity. Just to give you a warning, because we have seen that stated multiple times in the last couple of months. So FDA is expecting chemistry for novel gas path devices. That's in the document. And then we have changes to the table. So we go through this in a little bit of detail. I do want to talk about the big impact items to what I see from the FDA. This first one is this practitioner contact. When we talk about practitioner contact, a lot of people get freaked out because they're like, man, my surgeon, he touches the handle of my device, and I've never tested the handle of my device. So now do I have to start assessing the handles or whatever it could be contacting the patient? In the document is specific for type of contact that is not protected. So like the doctor does not have PPE. The one that we just did recently was a headband for a face shield of the surgeon. So the headband contacts the surface of the skin, right, of the surgeon, and so he doesn't have gloves or a sheet protecting him. So we had to assess the biocompatibility of that headband. That was actually asked for by the FDA. So they submitted, the FDA said, well, we like everything here, but the headband you didn't assess. And they're like, well, that doesn't contact the patient. And they stated that in their new guidance document, it has practitioner contact. So remember that. So if the surgeon has gloves on, then you do not have to assess the handle for biocompatibility. But if they're contacting the practitioner skin, then we'll have to assess some kind of safety. Okay? Once again, that doesn't mean testing. I want you guys to get that out of your mind. When we assess biocompatibility, it doesn't mean testing. It means assessing the endpoints and evaluating risk. Okay? So with that, talking about risk, we talked about the plans. I do want to tell you about what the biological evaluation plans entail. We really go through the materials that contact the patient and the processing of the device to really understand what is coming in contact with the patient. If we can do that just by a material and processing review by itself, then we should be okay to assess risk. For some devices, when they don't understand what's in their materials or don't understand what's in their processing or could be on their device from a process, then we have to move into chemistry to help evaluate that impact. Okay? So chemistry plays a more important role with your upfront risk evaluation, but it also can be your friend to help eliminate testing on the back end. So these are the things we put in the plan. Remember, the plan is what we do up front to evaluate risk before you start testing. Then you test to evaluate, to mitigate that risk, and with your BER, you sum it up in the document. Chemistry is here, okay, all through this document. And I want to tell you right now, ISO 10993-1 will be released hopefully next year. There's some discussions on maybe delaying it for 17-18, but hopefully we get it next year. I want to tell you, -1 has chemistry written all through it, okay? If you're not used to doing chemistry or talking about extractable leachables, you better start getting used to it because chemistry is now becoming biocompatibility. It's just another way to assess biocompatibility. It's a great way to assess biocompatibility because it's not just a pass-fail test. We really get to understand what comes off your device. Now, my personal opinion, if you have a low-risk device like a surgical tool or something that's in contact with the body for a short period of time, chemistry, even though I love it because it gives you good data, from a financial point of view, it may not make as much sense because you're just looking at the big three, right? The cytotox, the sensitization, irritation, maybe acute systemic tox. So maybe you just do those tests. But if you have anything that's prolonged or permanent contacting to the body, chemistry may become your friend because it can help eliminate some of those more expensive and long-term tests. So this is more for your reference. -17, ISO 10993-17 and 18 are the standards for the chemical characterization. -18 is getting completely rewritten. I mean, like thrown out the door, and a brand new document is being written. Okay, so that's going to be a lot more help to the industry going forward. Attachment C. This is one of my favorite things to the new guidance document. So they have added an attachment C, and what attachment C is, I think the FDA was getting tired of getting all these different ways of submitting biocompatibility data to their agency. I mean, think about it. If you're a reviewer and you get a brand new document, how long is it going to take you just to go through and try to find where everything is, right? So they've released attachment C, which is how to summarize your biocompatibility in a way that they're familiar with it, okay? So they actually give you this chart. Okay, so this is how they would like to see the information provided to their reviewers, so that it's consistent. It's something that they're used to seeing. So attachment C in the document, this is actually the chart out of attachment C, so it can help you understand how they would like the biocomp information provided. Remember, you still have to provide that BER, that biological evaluation report, which helps sums up all that stuff too. We talked about cytotoxicity extracts. For me, I think this might cause a lot of devices that have previously passed cytotox to maybe have a higher elevation and now have to evaluate why they're starting to see higher cytotoxicity, and is it really clinically relevant, which is a question I ask every time I see a failed cytotox. Is it clinically relevant? We're making the most sensitive test more sensitive by doing this. How much do we really need to depend on it? We talked about complement activation. We talked about implants, and then implant devices must consider that LAL test now. Okay, big impact for orthopedic or really any implant device that didn't previously had to do the LAL test. Now we talk about the new table, and for me this may be the biggest impact. In fact, we are going to be changing these, so next year come back and get a new one because we're going to be updating it to this new table. It really hasn't changed that much. Okay, if you look at it from the point of view here, it looks about the same thing. But the big changes were they added a material-mediated pyrogen section. Before, we kind of assumed that if you had acute systemic tox marked, you may want to look at material-mediated pyrogen. The FDA now has put a whole new column in there where it talks about you have to do material-mediated pyrogen or assess it if you have that category marked. They also put emphasis on reproductive toxicology and degradation tests. They do say that you only have to do these tests if you think your device has a risk of these two endpoints. Okay? So if you don't have a risk of degradation or you don't have a risk of reproductive toxicology or toxicity, then you don't have to worry about those endpoints. But if you do, if you think you do, talk to us because we may have to assess those. But material-mediated pyrogen. And then the big one, carcinogenicity. Like I said, before this category was almost forgotten. Now the FDA is requiring us to assess that if it's an X or an O in that box. The other thing I want to mention is the new -1 that's coming out is also changing this chart a little bit. Before cytotoxicity, they're adding another column for material/chemical characterization. And every single thing is marked. When you see that, I don't want you to freak out that you think you have to do chemistry on every single device. But you do have to understand what your materials and your processes are for every single device. It's part of that risk assessment. So the new standard is going to have that column filled in, and every single category is going to have an X that you have to do material or chemical characterization. Okay? I think we have one more slide, and then we'll get to questions. So the main endpoint to this, the chart, so the chart split up into two sections because the FDA couldn't get it all on one page. Kind of frustrating. But the second page, the one thing I want to lay out is right there. So the FDA, if you look at our slide rule, has always had the circles in their G95. Those circles were tests that were just maybe considered necessary if the reviewer felt there was a reason to do the tests. Now, with this new chart, the FDA is requiring you to assess those endpoints, not just the X's, but the circles. So if you have a circle, you still have to assess that endpoint. Doesn't mean you have to test it, but we have to assess that endpoint. So the FDA is saying, look, if we're doing a risk analysis, and we're not saying you have to test everything, then we're going to make you assess those circles too. You tell us why those circles aren't needed. We're not going to tell you when they are or when they're not. You tell us why you think it. You know your products better than we do. Tell us why those circles are going to be needed or not needed. Okay? So, are there any questions? I know I just spit up all this information to you guys, and now I'm asking for questions. I wanted to give you all the things we saw in the FDA document. But like I said, if you want a copy of this presentation for your reference, just give me your business card and I'll email it over to you. But I would love to answer questions about anything biocompatibility related at this point. Anything grandfathered in? His question was, is there anything grandfathered in anymore? It depends on the risk. So we've been very successful in grandfathering in devices, but what we do is we evaluate the risk based on the history use of that device. So if we're looking at a device that, a good example is, I have a lot of companies who are coming to me now that used to make orthopedic implants. And they're like, we have this ASTM grade stainless steel that I have made implants out of for years. And I've submitted this last one, and the FDA is asking me for biocompatibility. He's like, I don't even know what that is because I have never had to do it. And so we sit down and we're like, okay, you don't necessarily have to do the testing, but let's evaluate, let's give you a risk assessment. And so we go through the materials, we go through the processing. Have you done predicate devices with these exact same materials and processes before? Can we draw risk based off of those? And then if we can, then we can help justify and write that plan together and then the report and submit that to the FDA. And we've been successful doing that if we have the information that's needed. So technically nothing is grandfathered in anymore, but you can still use the same concepts when evaluating risks.
So that means everybody has to go back, relook at their master files, relook at their compatibility data, and really do deeper assessments.
So his statement really is that everybody has to go back and look at their master files, look at their biocompatibility data, really do an assessment. My recommendation would be, yes, some kind of assessment. Now, if it's been on the market for 20 years, you haven't. Here's the other thing that frustrates me a little bit coming from a lab, is I hear this device hasn't changed. I have not met an engineer who can be with a device for 20 years and not change something, right? So just because the biocompatibility was done 20 years ago and it's been on the market, I would recommend some kind of assessment to evaluate what has happened, but that assessment may be it hasn't changed. The process hasn't changed. It's been on the market. There's no clinical issues. Then that's your assessment. But I would recommend doing some kind of risk-based approach to put into the file.
Or is it up to the companies and they're taking a liability risk? I would like to know.
So his statement was, is the FDA demanding it, or is it up to the company, and is the company taking a liability risk? Ultimately, the FDA is not, I haven't seen the FDA demand it, but I have seen audits where they say, where's your risk-based approach? And they say, well, we don't have any kind of risk based on this product because it's safe. And they're like, well, how do you know it's safe? And so at least have, even if it's just a page to go through the history of the device, where you've explained what happens, what the safety is, and that it's safe, I would put it in there because I wouldn't want that liability to have to put that product on hold while I go back and do expensive tests. So I would do some kind of upfront work. Yes.
How do you work with the startup companies in biomedical devices and biodesign companies? For example, Stanford University has a biodesign lab. San Jose State University has a biodesign education course. There are MS and PhD students who are coming up new devices and they need testing services. So how do you work with them?
Good question. So if you didn't hear, his question was, how do we work with like brand new startup device manufacturers that, and honestly, this approach sometimes hinders the brand new medical device manufacturer, because if I'm a very large manufacturer, think about it, I have a lot of materials that I can draw history from, a lot of processes that I can draw history from. It's easier for me to assess a risk because of my experience with materials and processes. If I'm startup, I might be pulling materials that I don't know really what they are. I might be looking for the cheapest materials possible. I might be subcontracting the manufacturing to somebody else where I don't have a lot of control. So it's a lot harder to do a risk-based approach, but not impossible. So we just have to know what questions to ask up front. I recommend that startups, anytime I talk to startups, get one of us, get a lab, any lab that you like, on the phone when you're designing up front. Have us tell you what kind of look for materials, what questions to ask your suppliers or your people you're working with to get the information up front. Because my experience is when they're trying to get your business, they're usually easier to work with than if they already have your business. I don't know if that's every industry, but that's definitely ours. So the other thing I do want to mention, real quick, be careful with colorants. The FDA did a webinar in February. Colorants, if you're adding, first off, if you have to color your device, pushback. Why does that device have to be purple? If you're going to sell 1 billion more of them, fine. But remember that the FDA is going to look at a colorant and go, especially if it's a prolonged or permanent contacting device, and go, is that colorant leaching? How much of that colorant's going to the body? Can you tell me how the toxicity of that colorant is? We can. There's ways to do it. It's just, is that colorant really beneficial to the use of the device? Is it worth all that headache? And that's the question you have to ask.
While the device is running, some insurance is discussing a duty to log in other industries. Has that yet impacted the medical device industry?
I'm not aware of any like duty log kind of impact. Larry, do you know any? No, I don't know any off the top of my head. I would not be surprised if something like that came down the pipe. But right now, they're just responsible for the safety of a device on the market, what kind of changes, what kind of impacts could have over time.
Are there any FDA requirements of devices about the device integrity against hacking?
Oh, against hacking. That's outside my expertise. I'm a material guy. So that's a great question. But I don't know if there's been anything put in. Do you have any idea? Yeah, so we're there, I'm sure there's concerns about hacking a medical device. I'm positive there is. It's just I don't get involved in that. So it's a good question. Any other questions? Yes. So that's a very good question. So his question, if you didn't hear, it's what's up with Europe, basically. Are they following the FDA? Are they still different from the FDA? Personally, my opinion, I've seen a merger between the two. So it used to be Europe was a lot less strict when it came to these things, especially when you're notified body auditing and things like that. They've had some issues over there with breast implants and some other things, and so they're starting to become more strict, like surprise audits, and really putting the notified bodies under strict control. The new medical device directives are coming out, regulations are coming out next year. They're going to be a little bit more strict, but the FDA has become more risk-based approach, where before they were more like just checkbox. So I think they've come together more, but a risk-based approach, especially with chemistry, will work in Europe and work in FDA. In Asia, right now, we're still maybe struggling with that concept of how that risk works, but Europe and FDA is still very, very good.
What if the use of your device potentially exceed the requirements for the 10993 code, and you feel that you need to apply more stringent criteria? What would those be? Is it still based on animal experiments, or you can do something more specific to basically assess for more chronic use of the product?
Yeah, so this is the whole reason we went to risk-based approach, was because this chart really can't envelop everything that's out there today, right? So if you follow this chart, but you're doing a risk assessment, and so if your risk assessment says, man, this degradation profile of my device is going to make this much more risky, then we have to set up either an animal test or a chemistry test or something to evaluate that risk. And so that's where that upfront work is needed, because if you submit it to the FDA and they evaluate that risk, they're going to have you have to do the testing, and sometimes those custom tests may take a lot longer to do. So we'd much rather have that risk assessed up front than having to try to respond to an FDA question. But yes, there's a lot of times where the product degrades, for example, or may have a new material or byproduct, something like that, that we may have to assess a different endpoint on this chart than what's in this category. Any other questions? So I could talk about Biocomp all day long. But if you guys do have any specific product questions, my booth's just 621, I think, just over there. So come by and see me, and thanks for your time.